Hello, welcome to one more lecture on the fundamentals of biomedical applications of quantum science. Last lecture, we were discussing what we call magnetic resonance. And we explained that we can have, due to the presence of the spin on the electrons and on the nuclei, we can have two types of magnetic resonance. The electronic magnetic resonance and the nuclear magnetic resonance. And the essential part of the magnetic resonance is the following. We apply a bias field, we open up the components of the spin of the nuclei, for instance, and like hydrogen, and then there is an energy associated with these two levels. I took the example of hydrogen, which has one proton in the nuclei. Therefore, and the proton has the spin half and has two components that will interact differently with the bias field that I apply. It's like having two different magnets with opposite polarity each state. And therefore, the interaction, the energy will grow. One of the components of this, one of the projection of the spin will grow with the field, and one will decrease with the field. At the end, applying a magnetic field, we have a split. We have like a two-level system. And then when we shine here, electromagnetic radiation here on the range of a radio frequency, this can interact with this two-level system because this is two-wave function. This is two-state, and what the radiation will do is mix that as happens for the electronic states that we discussed in the two-level approximation that we did. And as a result of this, we can make transition from one state to the other and vice versa. And therefore, if I send the electromagnetic radiation and I wait, I send a pulse, part of the energy in the electromagnetic radiation may be consumed, may be used to promote this variation of energy on the spin state. And that's a very powerful spectroscopic technique. Right, and I remember very clear that I showed to you these effects and showed to you how a spectrometer works, but I'm going to identify again NMR spectrometer because depending on the type of nuclei we have and the environment, we have absorptions. And as a result, we have applying the magnetic field, we can make a source, a transmitter of radio frequency, inject into a cavity where there is a region that the magnetic field is present. And normally we need big fields for that because, as you saw, and I explained last lecture, if I'm going to do the nuclear resonance, I need many Tesla of bias field, and the radio frequency will be will operate in a frequency of about 500 megahertz. And remember, for the electron, we go to many gigahertz. So I come here, I send the radio frequency, and I observe how much is absorbed. What is the quantity of radio frequency that's absorbed, and then I have a spectrum like we do in many other spectroscopic techniques. We measure the absorption, and we can identify what is the spin that absorbs. And what is interesting here in the nuclear magnetic resonance is that a given nuclei it will absorb specifically some frequency of radiation. But there are a few things that modify this frequency. Because, we don't have the nuclei alone. Normally it is surrounded by electrons, it is surrounded by other nucleus, and then there is a kind of local field that makes a shift on the frequency. It's like when I apply the magnetic field, I modify other things a little bit. And the way that this modify depends strongly on the environment of that nuclei. So that nuclei present there, right, in the environment has a specific frequency. So normally one type of spin will have a traditional frequency, the way I explained to you. But now, depending on the local field for a given nucleus, we have a local induced electronic orbital momentum and everything due to the existence of many electrons that has orbital momentum and that contributes to the field, and they respond to the field in a different way. At the end of the day, because of this local environment, we end by having a shift. I apply a field and there is a shift on the field. There is a tiny variation on the field. What the spin really feels is a field with a variation due to the local part. Of course, that's depending on the bias field that induces this variation. And that's dependent to a constant, and this is the constant that will somehow be very peculiar, be characteristic of the local where this atom is localized, right? That's called shielding constant. Therefore, what's happening is the total field seen by the spin of the nucleus at that specific position depends on the bias that I applied, that was things that I explained to you. And let me tell you, when I explained the basics of the magnetic resonance to you, I was being very general, having an atom isolated, so looking that nucleus of the atom without any other interference. But now, if I'm looking one specific atom inside a molecule or inside a solid, there will be a local contribution. And this is given by this shielding. So actually, because of this local response to the main field that I apply, the overall field that the spin of the spin component of the nucleus, what they really feel is a composition of the field I apply and the local field variation. So normally we write as 1 minus sigma times B0. So there is a kind of characteristic way, depending where, look, let's imagine an atom of carbon. I can have the carbon alone, so I have a resonance. Because when I apply a field, it feels totally my field. But now I have a carbon inside a organic molecule. Well, then there is another environment, which is very different from the carbon isolated. And then I have this shielding effect. Because normally the presence of electrons try to respond to a bias field that's applied. There is a coupling between the properties of the electron distribution and the magnetic field that's applied. This is well known in atomic and molecular physics. Okay, so, and therefore, if I am smart enough, I can identify. I'm always looking for hydrogen. But depending on the environment of the hydrogen, I can know if the hydrogen is isolated, if the hydrogen is bound to oxygen or bound to a carbon or whatever. Why that? Because there is a different local contribution to the field. And because of the overall field that's filled by the nucleus, depending on this local, the frequency, as we discuss, the frequency basically depends on the bias field. But the bias field, not the field that I apply, because here I had the nuclei isolated. I had a nucleus that was alone with the radiation and the B field. But now, what I have is the nucleus in the presence of the bias field, but embedded in the molecular region, in the local field region produced by whatever is the chemical environment that this guy is there. So, in conclusion of this, the local field, which means the applied field plus the response of the local environment of the specific spin that I'm producing here, can help me to identify many things because the frequency will be shifted. So that gives me a chance to look into the same kind of nucleus, observe different environment. And that's the basis of the spectroscopy using nuclear magnetic resonance. So the caused shift, right? There is a shift in frequency that can be many megahertz. This is, I put like, I explain to you what causes the shift, and then explain to you here that this shift can be big, right? Depends on how is the delta sigma, how is the variation. Depending on the environment for each of the spin that I'm looking, I can identify different peaks on the absorption because of the modification of the frequency by the local field. Okay, so let's take an example. I know this, okay, well, now you can see well. I'm going to look at the ethanol, important molecule that we have. And I'm going to look at the NMR spectrum of the hydrogen in this molecule of ethanol. If you remember, ethanol has different possibilities. Ethanol is, let me write the ethanol here molecule, so it will be better too. Is a C, ethanol is C, H3, CH2, OH. And it has the two carbon and everything hanging around, oxygen, the OH, and the hydrogen hanging around. And then there is a possibility, if I'm going to observe the spin of hydrogen, of the nucleus of hydrogen, I have three possibilities. This is one hydrogen, this is another hydrogen, and this is another hydrogen. Of course, here they are equivalent. So I call this hydrogen here, I'm going to call hydrogen 1. And when I look at the peak, this is basically the frequency. I'm putting here the frequency of the spectroscopy of NMR, and I'm seeing where the radio frequency is disappearing. So the first peak comes here because the shift of this is much less than the shift of the others. If I look at this one, which I call 2 now, I see this peak here. This peak is due to the hydrogen, still the hydrogen, but shifted by a few megahertz. Here, a fraction of megahertz. And if I look to this one that I call 3, hydrogen 3, I see other peak, which is displaced even longer, even further, right, in frequency. So, by looking at a spectrum like this, as a function of the frequency, I can identify. So, if I suppose I don't know that I have ethanol in my system, but I see these 3 peaks in the right proportion, I can identify, oh, this has ethanol in it. And more than that, I can find by looking to I can have standards of concentration, and then I can even find what is the quantity of ethanol in my sample just by looking this collection of these 3 peaks that identify the molecule of ethanol. Or normally there will be other peaks if there are other molecules with different with more hydrogen and things like this. If we look to what we call the hydrogen resonances, depending on the position of the hydrogen on the molecule, on the group of the molecule, we can identify many different shifts. So if the hydrogen is in the OH, or if the hydrogen is bound in a different way, is more bounded to the carbon, or if it is the hydrogen closer to a double bond, and so on, depending on if it is a hydrogen like a CH3 and then a radical on the other side, whatever is the hydrogen location on the molecule, I will have a different shift. And then I can identify many hydrogen. But I can do that with the carbon as well. You know, the carbon is a nice nucleus and has many resonances. And depending on the position of the carbon and the presence of a pi electron close by, or only sigma electrons close by and everything, I can identify where it is. So NMR, nuclear magnetic resonance, is a very powerful spectroscopy technique. Right? And we can have more example if you want. Right? I can give you more example in different molecules, always looking to the same transition, but in different environment. So this is a real spectrum of a molecule, an NMR molecule, an NMR spectroscopy of a more complex organic molecule, and we are looking what? We are looking hydrogens. But we have so many different hydrogens. Or we could be looking carbon, and we have so many different carbon that we can identify. Or even oxygen, we have the peaks. So normally looking the collection of the peaks on the NMR, we identify not only the molecule, but we can identify, suppose that this peak here, right, this peak here, this peak here is shifted a little bit. So we can identify that something stick closer to it because it changed the shift, because it changed the environment. So we have many numbers for carbons or for hydrogen, and we can identify the NMR. I don't remember exactly if this is for the carbon. I think it's for the carbon. So this is the carbon type of the carbon type of NMR. And we can identify a sequence of peaks, and through these peaks we can identify what carbon is contributing to what, and we can identify the molecule, and we can identify variations of the molecule. We can identify if it is bounded to a surface, what is the molecule doing, just looking to the spin resonance. That's a very nice application of the NMR. And the quantum mechanics, because this is all quantum stuff. Nothing is classical there. But there is a nice application of NMR, which is the more popular kind of application that everybody already hear about, which is the magnetic resonance imaging. Can we really, by mapping some molecule, do imaging? Normally this is done with hydrogen because it's a very good signal, and in principle we can do wonderful things by looking, and because it's dependent on magnetic field amplitude, we can do a lot of things, including look inside the body or looking inside an object. And this is the I wrote here that's the most striking application of the magnetic resonance, and it's very important for diagnosis. So we are able to look inside the body. We are able to see things that normally even not X-ray can do it. And we can do three-dimensionally, because you're going to see that I can produce gradient of field in one direction that I can put in the other, or cycling gradients of fields and then map the resonances that I have. That's very important. So let's see how this works. How magnetic resonance imaging works. What is the principle? Well, the principle is here. You know, we could write the principle here. What is the principle of this quantum application to see things in biomedical? Well, a magnetic resonance extrapolates the biomedical. You know, you do magnetic resonance to study soil, to study fossils, to study materials, to study anything you want. The principle to do the imaging using nuclear magnetic resonance is the following. Imagine that different parts of a tissue have a different constitution and therefore different concentration of protons. Right? I have more one type of molecule than the other, and so on. Using this fact, using the fact that different parts of the tissue have different concentration, I can do a spectroscopy in each little part and then compose this part with the right contrast and make an imaging of it. Actually, what we are imaging is the concentration of hydrogen. But hydrogen is so important for living tissues that depending on the concentration, we can identify different parts of the tissue or even a transition from one tissue to the next. The question here is, if we can determine the proton concentration or the hydrogen concentration in different positions of space, if we can do that, then we can produce a map. We make a map, like a map. The same way you make a map of a country saying where is the plantation of different types of plantation, production, or oil production in one state, and fruit production in the other, and beef production in the other, and so on. You do a map of the concentration of hydrogen. And that then, this map is a planar map. It's a map on the plane, 2D map. And then we compose the 2D maps, and we can create, you know, the topological of this thing. And that's the principle. If we can do that, we can do the imaging. The question is, of course, how to produce this spatial resolution, right? Because we have to map the quantity of protons, the quantity of hydrogen in tiny little different positions. How can we see hydrogen that are in different positions? Well, here comes what I teach you on the overall ideas of the magnetic resonance. And what I teach you on the overall ideas of the magnetic resonance is that if we have a sample that contains protons and we submit to a constant field, this field determines a fixed resonance. What about if I produce a field, different field in different parts of the space? If I produce a field that's different in different parts of the space, what I'm doing is differentiating the space depending on the hydrogen present. And I measure for each field the frequency and the absorption. So I can map without going there, but making sure I know the field at that position. I can do the magnetic resonance. And I can find how much hydrogen is there and in what conditions, taking into account all the things I told you already, the shift due to the local environment and everything. And that's what we do to produce the imaging, to produce the magnetic resonance imaging. So, see, to produce the imaging, we have to make a kind of mapping. So we have to distinguish between things that are in different positions and being able to measure different positions. But thanks to the magnetic resonance technique, we can produce fields specifically in each part of the space. Therefore, we can sample different parts of the space, varying the frequency. And by doing that, we can map the hydrogen everywhere and produce an imaging. Okay, let me explain to you in a little more detail what we're doing. Let's imagine that I produce a linear field, a constant gradient field in space. This is the space, and I produce a field that grows linearly with the space. I'm talking about the coordinate z. I produce a field that has a gradient that's called g here, g of gradient, of course. Each position on this field, if I throw hydrogen in here, for each position, which has a different field, I'm going to have a local resonance for the NMR. You agree, right? Because the resonance is dependent on the field. But now I have a field that is space dependent. Therefore, my frequency is also space dependent. The frequency is also space dependent. Suppose I throw hydrogen in here and I flash a specific frequency. I'm going to see a specific position absorbing that energy, because I send one that's resonant for this Z. I can change the frequency and choose the one resonant with this Z. And then I measure in different positions what is the concentration of hydrogen with all the different possibilities of hydrogen in local fields and everything, in local shift. The shift is, of course, is much smaller than the opening of the levels caused by the bias field, right? We already saw that. And the shift is proportional to the bias field. So I have all the things under control to be able to identify where am I sampling by changing the frequency, because now the resonant frequency is locally selected. If I select one frequency for a specific Z, I am only interacting with the hydrogen in that position. Therefore, I can tell everything about this position, and then I tell everything about the next position, and so on. So I can make a map. How is the distribution of hydrogen in here? Of course, we cannot forget that I can do equivalent gradient in the other directions, x and y. So I can map everything in z, then map everything in x, and then map everything in y. And I have a three-dimensional map of the hydrogen. By looking the absorption, this is important, looking to the absorption of the radio frequency of different frequencies that now depends on z, will be proportional to the proton concentration at position z. With all the shifts and everything, I can take care of it. So it's like doing, in this case, what I'm doing a tiny little slice selection. I'm coming here, and by selecting the frequency, I fix the field, and by selecting the frequency, I am sampling a tiny little slice of space. And that's what I'm going to do here. I'm going to slice the space in many slices by selecting different gradients in different directions and by varying the frequency of my radio frequency. And therefore, I will be able to have a three-dimensional map. Well, to be a little bit more didactic, let me do this in a cartoon, right? So the idea is, I have a B field, which has a gradient, and then I select the different frequencies, and I can map the absorption. So I do slice by slice by moving my frequency. The field is static. This is fixed. I'm not moving it. So I do slicing, and I measure, I identify each position. So I have a collection of spectrum for each position. I measure the absorption, but then I convert the absorption in concentration of hydrogen. There is a full relation with that. And then I come and I draw here how is the concentration of hydrogen as a function of Z. If I do that in Z, X, and Y, I create a 3D kind of picture of the concentration of hydrogen in the sample. And the field is so nice because for biological tissue, we are almost transparent to the B field, almost. Therefore, by imposing a gradient, I am sampling parts that are inside the body. And I can verify inside the body without hurting. Suppose there is an interface. There is an interface changing tissue. What I'm going to do is, as I sample, I'm sampling by slicing one side. When I move to this part, I'm going to see a jump. I am able to identify, by this jump of frequency, identify the interface. So I can see variations inside, tiny little variations inside one part, and I can see the interface, tiny little variation in the other side, and so on. Therefore, by applying the slicing technique in different directions, I can produce a 3D imaging. And that's the basics of using the quantum mechanics, using basically two-level interaction with radiation. Here, the spin components, I can map inside the body and make a wonderful picture what's going on inside. Well, I mean, everybody has seen machines that do that. And I want just to point out for you here that the machine normally has the main magnet, which is the magnet that produces the big field. And then we have shim coils to equilibrate, to really knows the field well inside. We have the gradient coils, and then we have the radio frequency coils, the one that illuminates. And then we put a person inside. We know very well the map of the magnetic field, and then we move this person inside. We know exactly the position, and we apply gradient in different directions, do the resonance, do the NMR. So this is an NMR machine. And by doing, I'm able to map the whole body if I want, and then identify diseases, or identify brain activity, identify all kinds of things. Normally it's big because we want to do big persons. And because we need big fields, many Tesla, as I told you. Normally there is superconductor magnet involved to improve resolution, stability, and everything else. What kind of output I get from this? Like this, right? You'll be seeing this in many, many places today. This is kind of everywhere. Because I'm able to scan what I did here. You can imagine that in a plot like this, I was scanning this direction. And I made the gradients, and I was, producing images on each Z here, if I call Z, and then I moved to different positions. Or I did the gradient going that way, and then I mapped this way. Doesn't matter how you did. When you combine it, and then you need some very nice software to combine, you can see inside the brain. You can see many things inside the brain. What can we see inside the brain? Well, you can see tumors. That's a tumor that's in the brain, and I could identify well, but I could see many, many things, and in fact, I can even see what the tumor is affecting, because the guy is thinking all the time, and then I can see. We can see what they call the activity during body movements. We can see what part, because when the brain is working, there are energy transfer, there are tiny chemical variations of the environment and everything. And then I can see where is the activity of the brain. You can observe in this, depending how the guy is moving hands around, I can see part of the brain becomes brighter in this mapping, right? And that is moving, that region is moving around depending what's happening, what kind of activity, what kind of emotion, what kind of everything. And then I can understand how the nervous system works, the central nervous system works, how the brain works. And then I can do many, many, many wonderful things in science and biomedical engineering just by understanding that kind of thing. So you see, NMR is a big revolution in diagnostic of humans and animals and anything. Even plants, you can see that a tree. You can come and do an MR in a cross-section of a tree, and you can identify the age, you can identify if there is a empty part in the center that will compromise the tree stays up, and so on. An MR is one of those resonances that quantum mechanics gave to us because of the spin property of the protons. That is wonderful. And of course, to do this, we had to know the quantum mechanics. Otherwise, we will be unable to understand how to produce those resonances and how to use it, right? So you see that quantum mechanics is already giving us many, many things. We are surrounded by quantum mechanics, and we are surrounded by techniques that is coming from the understanding of quantum mechanics. But NMR imaging is not only for humans. NMR imaging is a powerful technique that's being employed in many, many, many other areas. And I just want to highlight once. Physics, of course, is very important in physics. Is one of the techniques that help us to understand a lot about physics and even improve the quantum mechanics itself. But there are many other applications. Soil science. You can verify constituents in the soil. You can do all kinds of things in soil. You can see porosity in soil. Because or stones, right? If there is oil inside the soil, that's very important for states that explore oil all the time. Use NMR to identify the quality, the porosity, quantification of the quantity of oil inside. But so soil is important, but it's not the only. Agriculture, the fine agriculture, to understand how the plant developed in terms of the quality of the soil and everything is a big application. Medicine, I just give you examples in medicine, which is the most important for the biomedical. Hospitals, many different things. Functional imaging, as I told you, in the brain. Airports, detection of things that they should not be there. In vivo spectroscopy, this is really great and still developed, it's being developed. Microscopy, you can do NMR in a very tiny little resolution that allow you to really understand almost on the micro level what's going on. Food technology, material science, many, many material characterizations is done by NMR. Structural biology, molecules, proteins, DNA, anything that's related to do structure biology. Structural chemistry, which is also important. Sometimes they don't need to do structural of biomolecules, but other things. And forensic science, which is important in a world that has so many crimes. We have to have techniques that allow us to somehow look to this. And there is one application that NMR is being used for quantum computation as well, which I left behind because I want to concentrate more in the life science type of applications. So you see, quantum mechanics is a wonderful technique that allow us to develop things like an MR imaging, an MR spectroscopy. And that's not only the technique, it's everything that the technique can promote. Can you imagine a world now without an MR? It's very hard, because we'll be a little behind on the development of materials, on the diagnosis, on brain surgery, and everything else. So, you see the value of quantum mechanics when we come to the applications. Okay, this is one more lecture of this basis and application of quantum science for biomedical. And we still have 1 or 2 more lectures where I'm going to explain some wonderful applications of quantum mechanics on the molecular level to really apply for engineer application. See you there.